Welcome to Azad Bioacademy. Today I will talk about neoplasia, neoplasm, tumor, root causes of the tumor, cancer, and finally the characteristics of the tumors. Okay, so let's get started. Like neoplasia. So neoplasia means if you just try to find out the root word of the neoplasia, then you will get two things. One is neo, another one is plasia. So usually neo means neo and plasia means growth or you can say formation. So new cell formation, in a sense you can say a normal parent cell can be divided into new daughter cell. It's a normal phenomenon. But new daughter cell means not like unique cell. There is the identical cell. Normal cell will divide it into two cells and that must be identical to the parent. Okay, it's very usual process. So this is the mitosis process and this is the parent cell and this is the daughter cell. But this is not the new cell. You can say this is a fresh cell or daughter cell. But when you say new cell, that means structurally and functionally that would be different from the parent cell. That is the new cell. Okay, so neoplasia means new and plasia means growth. That means new cell or tissue growth. So if you just combine the neoplasia, so you can say there is new tissue growth or you can say formation. So what does it mean, the new tissue or new cell? That means the new cell, completely unique and different cell will produce that is not identical to the parent cell. How is it possible? That would be the question. That is how, and you can say why is it possible. Okay, so the normal parent cell will turn into daughter cell as per the demand of the neighboring cell tissues. Is that normal phenomenon? So what will happen when a normal cell, normal cell carry a particular brain of the cell, or you can say head of the cell, that is the nucleus, and you can say the brain of the cell, that is the genetic material. Okay, so when your brain will be normal, your body function will be normal, your physical appearance will be the normal. But when your brain structure will hamper, then your body function will hamper. So like that, when the brain of the cell, that is the genetic material, the genetic material of the cell is within the chromosome. A cell usually carry, the 23rd pair of chromosome, and chromosome carry the genetic material. And the main genetic material is, the DNA, and part of the DNA can be called as a gene when it will be responsible for the particular function. Okay, so when the genetic material will be altered, then what will happen? Altered gene will produce, and that will produce altered protein, that is faulty protein, or you can say this is oncoprotein. And that protein, faulty protein, will hamper the overall entire function of the cell, and that will produce a new cell. That means new cell means structurally and functionally different, new. Okay, so that cell will just gain a unique function. Okay, so normal cell has a normal function, normal cell, normal function, it will be functioning to produce energy, and cell will tissue, and the tissue will form organ and then organ system, your body is just functioning normally. But when the genetic material will alter, and if you just bypass all the defensive or all the correction process, then it will turn into new cell that will carry altered genetic material, and that will produce faulty protein, and the faulty protein will just show the altered or different function other than normal. Okay, so it will gain a new function. It will gain a new function. That means it will gain a new function. So this is the new cell. When such type of cell production will be uncontrolled, an abnormal way, so that would be the neoplasia. Okay, so what is the main root cause or hallmark of the genetic material alteration? You know, this is called mutation. Mutation. So mutation is the main cause to alter the genetic material. You know, the alteration of genetic material is called mutation. That can be by external factor or by internal factor. Okay, so when your internal factor, the blueprint or your defense system will fail to detect it or destroy it, your apoptosis process will fail. That can turn into the abnormal cell, or your external things that will radiation, different types of chemicals will just make the genetic material alteration, and that will bypass all the mentioned process, and then it will turn into altered cell. That is the new cell, that is the neoplasia, new tissue or new cell formation. That is the neoplasia. And what will happen when uncontrolled abnormal cell will produce uncontrolled way? Then that will produce a neoplasm, or you can say tumor. Okay, so neoplasia is the process and neoplasm is the outcome. That is abnormal tissue mass. You can say swelling of tissue. This is the tumor. Okay, so the tumor will form by uncontrolled abnormal cell division, and the root cause of is mutation. So when mutation, you know, the mutation is the matter of genetic material alteration. So what will happen when genetic material will alter, and how many ways the genetic material will alter, and what will happen in cellular level? Then we can categorize into two ways. One is genetic factor, another one is cellular factor. That will produce two types of tumor. That is neoplasm tumor, and that tumor can be two types. One is, you know, benign, and second one is malignant. That means benign means safe, or you can say innocent, and malignant means, you know, dangerous, or you can say invading. Okay, so how it will be malignant and why it is called malignant, why it is benign, how we will explain in two ways: in molecular level, genetic material related matter, and the cellular signaling factor or signal transduction related matter. So now come to the point that to find out the root cause of the neoplasia, that means tumor. Okay, so what would be the root causes? So already I already mentioned in two ways, that is genetic factor, another one is cellular factor. So firstly I will go for genetic factor. So genetic material, genetic factor, firstly gene related matter. So one is mutation, you can say oncogene formation, activation, that is matter. That means within your gene The genetic material, the genetic material is made up of nucleotide, and nucleotide is made up of phosphate, painted sugar, and the nitrogenous base. The main thing, the arrangement of nitrogenous base, adenine, guanine, cytosine, thymine. And the three of them will produce a codon, and that can specify a specific amino acid. So when the genetic material, that is your nitrogenous base, will alter, the overall function of the DNA will be altered. So mutation or oncogene function. That generally in our body gene is wild type gene. Wild type. You can say also it is called proto-oncogene. Okay. When it will be mutated and it can turn into oncogene, and that oncogene will produce oncoprotein. You can say also called faulty protein. That is one of the major culprit to alter the cellular function. Okay, so number one, genetic matter is oncogene matter. So when it will be oncogene, normal gene will convert into oncogene. It has some major character. Oncogene. So I will explain in later on. So oncogenes means cancer causing gene that can cause cancer. Basically, it will focus on the malignancy. Okay, so mutation basically in a gene, it has several parts: promoter region, silencer, enhancer, or you can say transcriptional binding site. When such important site of the gene will be mutated, then it can easily turn into oncogene, and the function of the gene will be very harmful, and that will cause cancer or malignancy. Okay, but what will happen for benign tumor, I will explain later on when I will just differentiate benign and malignant tumor. Okay, so now come to the second point, the mutation of the gene, that is oncogene. And now number 2 point is, I will say, the loss or mutation of tumor suppressor gene. Okay, tumor suppressor gene. What is the matter? Actually, within our cell, 23rd pair of chromosome, and some chromosome carry a particular gene that can be a guardian gene, that can be police gene, that can suppress the unwanted cell division, that means suppress the tumor. So those are called tumor suppressor gene. So if I just give an example like P53, as gene location is chromosome number 17, and if you think about retinoblastoma 1, you can say APC gene, and cyclin dependent kinase N2A, and different types of tumor suppressor gene are there. And when these are the controlling gene that will suppress the tumor of formation. So if that gene will be mutated, then what will happen? The controlling material, if suppose if you think your guardian within your house, if it is just not functioning or is mental retardation, then what will happen? The whole family will suffer a lot. Okay, just like that. Now I just say some gene, like you can say SRC gene, you can say MYC gene, you can say HER2 gene, SRC gene, like that. Different types of oncogene. And here is the tumor suppressor gene. That gene, when it will be mutated, then overall cellular tumor formation will be hampered. So this is the second point of the genetic matter. Third one is gene amplification. Okay, the gene can amplify as per the demand of the cell, but when it will influenced by external way, by the intuition, then gene can amplify more than normal way. Gene amplification means gene will copy more and more unnecessarily. So this is the gene amplification. So it is one of the region of the formation of tumor. Okay, the fourth one you can say the hyper hyperchromasia. That means your chromosome will be more. That means your DNA amplification and your chromosome number of chromosome will increase more and more compared to the cytoplasm. So this is the hyperchromasia. Fifth one, you can say the rearrangement of chromosome. Rearrangement of chromosome. What will happen? The 23rd pair of chromosome. So one chromosome, one genetic material can transfer from one chromosome to another chromosome. One part can exchange. Okay, so it can be in two ways. One is translocation and second one is inversion. So in that way, a genetic material of the chromosome can exchange or can translocate. So this is the rearrangement of the chromosome. That can also be one of the major genetic cause of the tumor. Okay, and sixth one, you can say the epigenetic matter. Epigenetic matter. What does it mean? The genetic materials is arrangement of nucleotide, that means already says that the base, nitrogenous base. But here the DNA is wrapped up by some basic proteins, that is the histone protein, chromatin. Okay, suppose this is the histone protein, and the DNA is like thread-like substance, so it will wrap up of that base protein. So this is called histone protein or chromatin. So that protein will be altered, will degrade. So histone protein can be degraded or can be altered. Also, the DNA can be methylated. So this is not the basic main alteration of the genetic material, but you can say this is the alteration of the supporting material of the DNA. Okay, so the alteration of the supporting material of DNA. That can be methylation or that can be histone protein degradation. So this is the epigenetic, not directly genetic. This is epigenetic matter. So in that way, a gene can be altered, gene can be mutated, or gene can be different functioning. Gene can be different than normal to produce the malignant tumor or benign tumor. That means that can influence the uncontrolled cell division and the other factor that will explain in cellular process. Okay, so this is the just mind it to find out the benign and malignant tumor. You can also focus on the major causes of malignancy or cancer. Okay, so these are the genetic matter. Now I will go for the cellular matter. What would be the cellular changes of the mutated gene? Okay, so cellular changes. One is you can show the signal change. Signal transformation change. Like when a mutated gene will produce oncoprotein, or you can say faulty protein, by the causes already I explained in genetic factor, and that will work in different way. Like here is the cell, it will produce within the cell, and it will transfer from inside to outside, and it will bind with the receptor here, and that will again increase the signaling pathway, and that will activate the genetic material. So that is the autocrine process. Autocrine process, it can work as a growth factor. That means oncoprotein or faulty protein can act as a growth factor, and that can work by autocrine pathway. Also, you can say that oncoprotein can just come out and work on the neighbor cell. This is the paracrine process. Okay, here the oncoprotein or faulty protein can influence or can activate the normal gene to produce more and more, and that will increase the growth factor. That means faulty protein or oncoprotein can act as a growth factor, and also it can overexpress, it can influence to overexpress the growth factor. Normal growth factor will also increase, and that will work again and again, and that will influence to proliferate the cell. So the signal transduction pathway can change by the faulty protein, and that faulty protein work directly as a growth factor, or it can increase the production of growth factor. That will influence the proliferation. Okay, so cellular, one of the major matter is the cellular one. Second one, I will say, is the you can say that is your anti-apoptotic protein production. And apoptotic protein or pathway suppression. That means the faulty protein can suppress the apoptotic protein production. You know, apoptotic protein is very much important. Already earlier I said the p53 gene, the guardian gene, that can produce p53 protein. That protein will activate the p21, and that p21 can inhibit the cell cycle, and also the p53 itself can act as a apoptotic protein, and that can cause apoptosis, that is programmed cell death. Okay, but here what will happen, the faulty protein can act as an anti-apoptotic protein that will inhibit the apoptosis process. Also, the anti-apoptotic protein production within the cell will increase by the mutated protein. Okay, that means anti-apoptotic protein, if I can give some example, like your BCL2, MCL1, these are the like anti-apoptotic protein. So anti-apoptotic protein means that will inhibit the apoptosis. Anti-apoptotic protein has some normal function. Within our normal range, it has some function. But what will happen for the tumor, that anti-apoptotic production rate will be more. Okay, and the apoptotic pathway will be suppressed. So what will happen? Ultimately, apoptosis will be inhibited. Okay, so what will happen? The programmed cell death, that means when faulty cell will try to replicate, then automatically the apoptotic pathway, intrinsic pathway, that is caspase pathway, will activate, and it will destroy the cell on its own. That is the programmed cell death. But in the tumor cell or cancer cell, that will cause the opposite things. That means anti-apoptotic production will increase, and the apoptotic pathway will be suppressed. By what? By faulty protein, or by the influence of faulty protein, the anti-apoptotic production will high, and the apoptotic protein will be less, so that finally apoptosis will be inhibited. Okay, so this is the way that it will make the path very easy to produce abnormal cell. So this is the cellular pathway of the three. Third one, it can be said the proteolytic enzyme production. Proteolytic enzyme production. So what is the meaning of proteolytic enzyme production? This is the, you can say this is the cell, these are the cells. So that cell is basically circulated, or you can say this is the covered by the extracellular matrix. These are the extracellular matrix. So that can be, you can say collagen, you can say elastin, you can say proteoglycan, you can say fibronectin, and so on. So these are the extracellular matrix, and that will just make a connection between two cells, and also you can separate the two cells in a certain extent. Okay, and the cellular adhesive molecule are present on the surface of the cell. Cellular adhesive molecule, that is cellular adhesive molecule, that can be cadherin, you can say integrin, you can say selectin. These are the molecules, and that the cell adhesive molecule and extracellular matrix are connected with each other, and the cell are located within the particular area, and that can be not easily translocated from here to there, and that will form a particular tissues. So identical cell will produce a tissue, and then organ, then system. Okay, so no way easily to transfer from here to there by the extracellular matrix and intercellular and the cellular adhesive molecule. This is the normal phenomenon. Then what will happen? For the tumor formation, it can happen, the proteolytic enzyme production will be higher, and in malignant tumor it will be different, but I will explain the proteolytic enzymes. You can say matrix metalloproteinase, that can be matrix metalloproteinase 2, matrix metalloproteinase 9. These are the proteolytic enzyme. You can say collagenase. You can say plasminogen activator. So different types of proteolytic enzyme are there, and that proteolytic enzyme will just degrade the extracellular matrix. And what will happen? The extracellular matrix will degrade, and the cell will attack the neighbor cell. Okay, the cell will attack the neighbor cell, and the cell will attack the neighbor cell. This is called invasion. So invasion is one of the major criteria of tumor. This is malignant tumor. Okay, the proteolytic enzymes, this is that will degrade the extracellular matrix. If you think it is the matrix metalloproteinase, in a certain extent that will be very normal phenomenon that will help to remodeling the tissues, but during the tumor formation, the proteolytic enzyme production will be higher. The matrix metalloproteinase, it can also be called the seizure protein. The seizure, it will just cut. So that is the proteolytic enzyme production for the cellular process that will degrade the extracellular matrix, and that will just facilitate the cell to invade the neighbor cell. Okay, so these are the cellular matter. If I just add another thing for the cellular factor is 4, you can consider another thing like angiogenesis. That will some factors that is endothelial releasing growth factor. That will, the production of endothelial releasing growth factor will increase, and you know the tumor means the multiple cell division, and you know the uncontrolled cell division. So here some different you know the cell division is more than normal. So what will happen? It will not follow the normal respiration metabolic pathway. It will try to produce its energy without help of oxygen. So it will produce energy, ATP, and also produce you know the lactic acid. So in the area of tumor formation is acidic because of the lactic acid production. That means it will alter the metabolic pathway. It will try to produce more ATP without help of oxygen. So angiogenesis and fifth one, you know, alteration of metabolism and different characters will show by the cellular pathway. Okay, so the proliferation, proteolytic invasion, and anti-apoptotic matter that will just facilitate the cell to produce more and more, and that will invade the cell, neighbor cell, and it will produce new blood vessel, and also it will just fuse the blood vessel or lymphatic system, and that will translocate from here to there, and that is called metastasis. Okay, it's one of the major criteria for a particular tumor, that is the malignant tumor. Okay, so these are the basic cellular changes, cellular factors, cellular matter, and some characters also already I said here. So now I will just differentiate two types of tumor, and then I will show the basic character wise difference. So now come to the difference between tumor, that is malignant, and benign tumor. So now here is benign tumor, and this one is malignant tumor. So here benign tumor, uncontrolled, abnormal cell. This is common. Both are same. Uncontrolled abnormal cell. Here the uncontrolled abnormal cell pattern is different, but the criteria or the uncontrolled abnormal cell division is same parameter of both benign and malignant tumor. Okay, and then if I go for particular difference, then I have to categorize the malignant tumor into by a particular parameter, that is loss of differentiation. Loss of differentiation, and that loss of differentiation means a cell will produce in abnormal cell or uncontrolled way, but in benign tumor the cell differentiation will as like as normal matter. That means a cell will be divided into two cells, and that two cell will be differentiated into different type of cell, or it will differentiate into two matured cell. But here the malignant cell, the differentiation will not happen. So loss of differentiation. And if you just define this term by a particular another term, that is the anaplasia. Anaplasia. So I will explain anaplasia in later one. Anaplasia will mainly reflect the malignant tumor. So I will explain neoplasia in later on. So now I will go for in different way. That is your cellular arrangement here is almost normal tissues like. That means it will be differentiated in a normal manner, and your cellular structure is as normal cell, somewhat different but almost like identical like normal cell. But here is different. Like if you think this is the normal cell, and you can say this is the benign cell and this is the malignant cell. This is completely different. And the arrangement of benign tumor is like almost like that. If you think in that way, these are the arrangement of cell, and here the arrangement is different, like that. Okay, it has. If you think this is the normal, so here some abnormal like that, but almost normal. Cell pattern, and the rate of cell division here, the nucleus size and the function almost like normal cell, but malignant cell is different. So I will explain by another term which is particularly for benign tumor, then I will skip it and I will go for the malignant tumor. So that is the encapsulation, or you can say capsule of benign tumor. That means when benign tumor will form, that most of the benign tumor, not all the tumors, benign tumors are circulated or enveloped by the encapsulation, or you can say capsule formation will happen to all the benign tumors. It's not correct, but most of the benign tumor are encircled by some fibrous connective tissue, fibrous connective tissues. Okay, how is it possible? Because when a cell will be mutated and it can produce abnormal uncontrolled cell, and that will, if that happens, then what will happen? Your neighbor cell will try to defend it. This is normal phenomenon. Okay, your body or cell, its own defense system, that will try to control themselves from the external influence or from any unwanted matter. Okay, so when uncontrolled abnormal cell division will start, then what will happen? The external, the extracellular matrix production will increase. That means your fibrous cell, that is fibrocyte, will be activated and it will produce more extracellular matrix, that is you can say collagen, elastin, or you can say fibronectin. And then what will happen? It will also suppress the proteolytic enzyme to encircle the uncontrolled abnormal cell. Then what will happen? It will make a circle, it will make a rigid fibrous connective layer, and that will act as a capsule. So this is very unique or identical for the benign tumor. That is not possible for malignant tumor, because malignant tumor will produce more proteolytic enzyme and that will degrade the extracellular matrix and no possibility to form the capsule. And I will repeat it, the capsule formation is the cellular or tissue's own defense system. It will try to form to tackle the uncontrolled cell division, and it will inhibit the spread out throughout the area. So that is the fibrous connective tissue formation. That means it will increase the accumulation of extracellular matrix, that is extracellular connective tissues, and that fibrous tissue will form a capsule, that is the fibrous connective tissues, and that is the different. So in that criteria, benign tumor is not easily invading the neighbor cell, or it can not express, it cannot spread out throughout the body. So here I can just add it. It has no invading active capacity. It has no any metastatic activity. Okay. So another point I will add here, that is oncogene. So oncogene, no, you know, oncogene means cancer forming gene. So malignant tumor must carry the oncogene, that is cancer causing gene, and that is basically mutated more repeatedly and frequent areas, that is your basic areas of the gene, like your promoter region, like your enhancer gene, like your transcriptional factor binding site, your guardian gene. Most of the prominent gene will be mutated here, but in the benign tumor, the less number of gene will be mutated, and the important area of the gene will not be properly mutated. And as like for that region, the benign tumor will act as like normal cell, and it will not spread out. Okay, so I will say the benign tumor can carry oncogene or may not carry oncogene, but malignant tumor must carry oncogene. And if the benign tumor carry oncogene, so a single question may come in your mind. So if it carries oncogene, then why it will not be cancerous? Actually, the oncogene, the extent of the mutation is very less, and the particular point, important point of the gene will not be mutated, so it is not properly producing the another factor that will the major criteria for cancer, like loss of differentiation, invasion, metastasis. These are the major criteria to be a malignant tumor or cancerous cell. So that criteria is absent here. So I will say benign tumor may carry oncogene or may not carry oncogene. Some mutated gene can show like a abnormal uncontrolled cell division, that can be the benign tumor, and it has some another criteria already I explained. Here all the criteria for cancer is present, so this is the malignant tumor. Okay, so I will explain the anaplasia matter, anaplasia mechanism, which is mainly applicable for benign tumor. So, sorry, malignant tumor. So I will just skip the benign tumor. Other very silly matters are there. If you just read from any site, you can easily understand, because already the background cause of the tumor formation already I discussed. So now come to the anaplasia character of malignant tumor. So anaplasia. Anaplasia is characterized by loss of cell differentiation. If a cell is lose its character, the differentiation, that means a cell will not turn into a different cell. So you know the tissue has multiple types of cell, and that will all together work in a harmonious way to form a tissue function, organ function. So anaplasia, you know, the loss of differentiation. Then different parameters I will explain, that is that is pleomorphism, means you know the number the shape and size of the cell will be different than normal cell. Okay, so already I said in benign tumor the shape size is almost identical to the normal cell, almost identical, but not not exactly the normal cell like. But in malignant tumor, the pleomorphism means the shape and size of the cell will be different than normal cell. Okay, another one is rate of mitosis. That means rate of the cell division. It is high than normal cell. It is more higher than normal cell, and you can say it is higher than benign tumor. So rate of cell division is less in benign tumor and more in malignant tumor. And I will say loss of polarity. It means the orientation of the cell of the tissue will be different, will be altered. That means already I said here, this is the very organized way of the cellular arrangement, but the cellular arrangement will alter. This is the loss of polarity. Okay, and that loss of polarity is very less common in benign tumor, but it is very prominent in malignant tumor. Another one is, the giant cell presence. You can say Bayesian cell. That means a peculiar or very giant or big large cell is present in the malignant tumor, but this is not common in benign tumor. And another point of the benign tumor is your angiogenesis, because new blood vessel formation. Already I said that will produce a endothelial releasing growth factor that will produce more and more blood vessel that will supply the more blood to the more abnormal uncontrolled cell, that is excessive number of cell. And then already I explained that is invasion. That means it will increase the proteolytic enzyme and that will invade the neighboring cell, and that will just encroach the neighboring area. And then metastasis. That means it will spread throughout the body by two ways. One is lymphatic way, another one is hematogenic way. Hematogenic pathway. That means by blood or by lymphatic system. Okay, so if I just add here for another point is hyperchromasia. That means excessive chromosome production, that is excessive DNA replication, or condense the nucleus, dark nucleus will be there. So this is not common in benign tumor. And another thing is that telomerase activity. The normal cell usually just take part in the cell division by 50 to 60 times, or some places 60 to 70 times. Because of why? Because the telomere, the upper top part of the chromosome, it will carry some repeated gene that will act as a telomere, and that telomere will control the cell cycle. What will happen? That when in normal cell, it can take part in the cell division by 50, 60, 70 times, and each time of the cell division, telomere will decrease. So telomerase activity is fixed or very negligible in normal cell, but in the malignant tumor, the telomerase activity will be higher. So telomerase activity will be higher in malignant tumor. Then what will happen? That telomerase enzyme will work on the telomere and it will prevent the reduction of the size of the telomere during the cell division. Okay, in that case, you can say malignant tumor, malignant cell or cancer cell is immortal. Okay, this is the way, because in normal cell, when it will go through the cell division, the telomere will decrease, but during the cell division of the cancer cell, telomere will increase. So cancer cell is immortal. So these are the characteristics of malignant tumor, and these are the factors that will actually, those are the features of anaplasia, and anaplasia one of the major feature of malignant tumor. So this is the difference between malignant tumor and the benign tumor. Okay, so I will just sum up the whole lecture by 2 minutes. That is, today's lecture is neoplasia. It means new cell formation. It does not mean new cell formation, that means new type of cell. Actually, this is the it's a new type of cell actually, not the fresh cell or daughter cell. The new type of cell, that means this is unwanted cell, undesired cell that will proliferate or that will produce more and more. That means uncontrolled cell division. And uncontrolled cell division, abnormal uncontrolled cell division will produce the tumor, and that tumor can be in two types. One is benign, another is malignant. Benign is safe and innocent, malignant is dangerous and invading. And it has some other character. Those characters are, you can say, invasion, metastasis, and you can say it has several negative sides, that is revive after removal. But here benign tumor is completely different. Benign tumor has some unique side, that is it has encapsulated matter, and actually benign tumor cell can be differentiated and it works as normal cell, but the uncontrolled cell proliferation is there, so it is some abnormal mass. This is the tumor. Okay, so benign tumor and malignant tumor, I will give some example here and then I will sum up. It is benign tumor, you can say fibroma, you can say lipoma, you can say osteoma, you can say adenoma. So these are the oma. Okay, oma is the common. Oma means tumor. But here, malignancy, you can add here carcinoma or sarcoma. That means it has also oma. Oma means tumor. So already I said mutation is the main cause of cancer, and you can also say the tumor is one of the main cause for cancer, but not all the tumor. So oma is the major matter. So when fibroma, then you can say fibrocarcinoma. Okay, fibrocarcinoma. So fibrocarcinoma, you can say adenocarcinoma. These are the malignant tumor, and this is the benign tumor. Okay, and if that tumor is so carcinoma, what does it mean? Carcinoma means the cancer will happen in epithelial tissue. So this is the carcinoma. And when the cancer will happen in connective tissue, then this is called sarcoma. Okay, so squamous cell carcinoma here you can say, and sarcoma, fibrosarcoma. That means sarcoma is connective tissue. You know the fibrocyte is the connective tissue part. So sarcoma can be there. So malignant tumor at the end, that is suffix of that tumor can be carcinoma or sarcoma, and the suffix is of the benign tumor is oma. Okay, this is the difference, and some exception also. If you think this is the myeloma, that is the cancer of plasma cell, you can say lymphoma, WBC, B cell lymphoma. So these are the malignant tumor, and this is the benign tumor. Actually, cancer means, you know, the benign malignant tumor, malignancy. That is the cancer causing tumor is malignant tumor, and you can say it can turn into cancer. So these are the character of cancer. So that will carry carcinoma or sarcoma, lymphoma, myeloma, like that, and benign tumor just like oma. Okay, so this is all about the benign tumor, malignant tumor, neoplasia, and the root cause and the characteristics according to the genetic matter and cellular matter. So this is all about that. Thank you very much.